This week we are going to be going over Chapter 3. Well, finishing Chapter 3. We started it last week. And then we'll also be going over all of Chapter 4, the selections that I put up. Just to remind you, Chapter 3 is all about the cell. The learning objectives is that you need to describe what a cell is and two general types of cells, and describe important landmarks in eukaryotic cells. Remember that only eukaryotic cells have organelles. And there are 9 important landmarks that I want to point out to distinguish eukaryotic cells. But first, if you remember from when I talked about the two different types of cells, eukaryote and prokaryote, which can be found in both eukaryotic and prokaryotic cells. So you might need to revisit those slides from last week. Okay, the answer should be ribosomes. So, number 1, the nucleus is only in eukaryotes, partly because it's an organelle, and partly because the DNA is in the eukaryote nucleus. The nucleus is one of the key defining features between a eukaryote and a prokaryote. Mitochondria, again, that's an organelle. Only eukaryotes have organelles. Number 4 would be the same thing. 3, ribosomes are not organelles because they do not have a membrane around them. So that's a defining feature of an organelle. Remember that the nucleus is the cell's genetic control center, and it is the largest and most prominent organelle in eukaryotic cell. I want you to remember that there are 2 primary functions. One is that it is the genetic control center, and the other is that it's the storehouse for hereditary information. And that hereditary information is called DNA, of course. And its DNA is stored in the nucleus in a form called chromatin. And chromatin is a mass of long, thin DNA fibers with proteins attached, shown here. So on the left, it looks like a bowl of spaghetti, but it's actually a bunch of chromatin all jumbled up inside of a nucleus. So that purple is representing the nuclear membrane. And then the middle is showing you that DNA helix is being wrapped around these histone proteins. So remember, histone. So DNA plus histone proteins equals chromatin. The reason it packs around these histone proteins we'll talk about later, but partly it's just so that everything can fit inside of the nucleus. Because each of your cells has 6 feet worth of DNA. That's a lot of stuff to pack into a microscopic cell. Not even the whole cell, just part of the cell. On the right is a condensed chromosome. So your cells usually don't have the DNA condensed like that unless they're about to divide. Usually the chromatin looks like on the left, like all jumbled up. But we'll talk more about that in subsequent chapters. So the take-home message is that the nucleus is large and prominent in eukaryotic cells only. It directs most of the cell activities because it is the control center and the storehouse for the hereditary information, the DNA. Alright, another part of the cell we want to talk about is the cytoplasm and cytoskeleton. They form the cell's internal environment and provide physical support and generate movement. So cytoplasm and cytoskeleton are present in prokaryotes as well. Cytoplasm is what forms the internal environment. It's kind of like a jelly substance. And then the cytoskeleton is what's providing the physical support and generating movement. So there are different types of cytoskeleton fibers, but I don't need you to know all of them. I would like to point out microtubules because those are tracks that help molecules move along inside the cell and get from place to place. But think of microtubules, intermediate filaments, and microfilaments as all coming together as sort of like building blocks to provide a scaffold. Just like if you were building a building, to maintain that structure, you need to put a bunch of wood together, and it's all different sizes of wood, or maybe you need some sheets of metal, and those are going to be different thicknesses or sizes of metal. And that's to help, number one, to provide shape and support for the cell. Number two, it controls the traffic within the cell. The cell is actually jam-packed full of stuff, and things have to get from place to place, so those microtubules are particularly important for number two. And then number three is cell movement, so allowing the entire cell to move. And that's shown here. This is near and dear to my heart. The cytoskeleton plays a more direct and obvious role in helping some cells to move. So on the right you see a organism with a flagella. So the quintessential flagella that you may have seen is sperm cells. They swim around. So the flagella is what's allowing them to swim, and that flagella is made up of microtubules. And then cilia on the left are short projections that often occur in large numbers on a single cell, and they can beat swiftly in unison, kind of like blades of grass in a field blowing in the wind. And their job is to move fluid along and past a cell. That can be important for sweeping the airways to our lungs, clean of debris, so that we can breathe. Okay, so cilia there's a lot of them on one cell, and they help to move stuff along, but not to swim necessarily. And then a flagella, there's only one or two for a cell. They're much longer and thicker, and they are going to help the whole cell move or swim. Here's the take-home message. Read on your own time. Next is the mitochondria. These are the cell's all-purpose energy converters. They're present in virtually all plant cells, all animal cells. Every single type of eukaryotic cell has mitochondria. A lot of people think that plants don't have them because plants have chloroplasts, but they do indeed have these. These are energy converters, so they're going to be abundant in cells whose job it is to use a lot of energy. So the muscle cells, liver cells, they can have a lot of these, up to 2,500 in a single cell. So we consume a variety of food, and our mitochondria is what allows us to convert the energy within the chemical bonds of the food we eat, such as carbohydrates, fats, and proteins, into carbon dioxide, water, and ATP. ATP is important because it is a molecule that is the energy source of all cells, used to fuel all cell functions and activities. We'll be talking more about ATP in Chapter 4. Because energy conversion in mitochondria requires a significant amount of oxygen, organisms' mitochondria consume most of the oxygen used by each cell. So in humans, for example, our mitochondria consumes as much as 80% of the oxygen we breathe. So pretty important for that. And they give a significant return on that investment, though, by producing about 90% of the energy our cells use to function. We talked about endosymbiosis before, but as we learned in our earlier discussion of endosymbiosis, mitochondria may very well have existed as separate single-celled bacteria-like organisms one day billions of years ago. That's because they are similar to bacteria in size and shape, and may have originated with symbiotic bacteria that took them up, and then they made permanent residence within the cells. Perhaps the strongest evidence for this is that mitochondria have their very own DNA, and it's circular, like in prokaryote DNA. And that DNA carries the instructions for making 13 important mitochondrial proteins necessary for metabolism and energy production. So we always say that DNA is in the nucleus, and that is true, like the vast majority is. But mitochondria and chloroplasts have their own little bits of DNA, and that's that's an evolutionary holdover from when they were prokaryote cells that were living unicellular lives. Another thing that supports endosymbiosis and makes mitochondria unique is that it's a double membrane structure. So you can think of it as a bag within a bag structure. So one membrane is a sandwich bag. You put that inside of another sandwich bag as another membrane, and you would have a two membrane bag within a bag structure. We're not going to go into why all of those membranes are so important, but they are. So in this graph, I wanted to just revisit, if you remember from chapter 1, what independent and dependent variables are. So take a second to answer this. And the answer would be the number of mitochondria per cell, or the mitochondrial number. And then which cell type in the graph might require the most energy? Alright, so different cells have different functions. That means different jobs. So some of them might need a lot more energy than other cells, so different organelles are going to be represented in the cells that need a lot of energy. Whereas other cells don't have very many at all. So the answer for this would be liver cell. So both liver and skeletal muscle require a lot of mitochondria because they require the most energy. But liver would be the correct answer, as shown in the graph. And then other cells like red blood cells, adipose cells, which just store fat, they don't really need a lot of energy, so they're not going to have very many or no mitochondria in it at all. So here's the take-home message for this. Figuring out how to approach a question in biology can be one of the most important steps in trying to understand how things work. In most cases, the best strategy is to take the simplest approach. Researchers wondered, for example, how an animal's cells might respond to exposure to a much colder climate. So they asked the question, can cells change their composition, or what they're made of, to adapt to their environment? So I'm showing you these slides to go over like an experimental approach, a practical application. So the way they set up the experiment to answer that question is that they use 15 cats from 5 litters. From each litter, 1 cat served as a control. If you don't remember what a control is, go back to chapter 1. So 1 cat served as a control while the others were exposed to extreme cold, minus 30 degrees Celsius. And they exposed them for 2 periods of 1 hour per day. The rest of the time, all of the cats were kept at 20 degrees Celsius. So one question you might ask is, why did the researchers use multiple cats from each litter? That would be reproducibility. If you do an experiment, you don't want to just do it on one subject. You want to make sure that your results are reproducible between subjects. So what they did after 1 week of treatment is they collected samples of the fat tissue from all the cats. And then they examined that fat tissue microscopically so that they could measure the number and size of mitochondria and the number of tiny blood vessels or capillaries per cell and the size of the cell. Okay, so after they exposed the cats to extreme cold or not, in the case of the control, they looked at their fat tissue and measured 1, the number and size of mitochondria, 2, the number of the capillaries per cell, and 3, the size of the cells. And you might be asking yourself, what was the purpose of counting the capillaries? Here are the results. So they're comparing the control cat and the cold stressed cats. And then there is a summary of the things they are measuring. For example, if we look at number of mitochondria in the control cat, you see that each cell typically has 1.48, whereas the cold stressed ones have more. They have 1.74. What that plus or minus sign means is how much we can believe that number. You can go back to statistics in chapter 1. So you want a really low number. It means that it's 1.48 plus or minus 0.11. So the number could be anywhere from 1.48 1.37 to 1.59. Anytime you do an experiment, there is a little bit of range of error. And that's what that plus minus sign is. That's the error rate. If it's low, that means it's a reliable experiment number. The size of the mitochondria between the control and the cold stress changes even more dramatically than the number of them. As you can see there, it also increases in the cold stressed. The capillaries also increase in the cold stress, and the cell size itself went down. The researchers used a single manipulation, exposure to cold, and then observed the consequences. This made it straightforward for drawing conclusions. Cold stress caused a change in fat cells. In the animals exposed to cold, the mitochondria became more numerous with each tripling in size. The number of blood vessels doubled, and the fat cell size was reduced to less than a quarter of the size found in the cells of the control group animals. So why do you think the fat cells in the cats exposed to cold shrank so much? And what change in the study would increase your confidence in the conclusions? So these are sort of questions I want you to think about anytime you see a research study conclusion. Essentially, it appears that cells in the fat tissue of cold exposed animals upgraded their heat generating system, and by making use of stored lipids to fuel the intensive heat production, they depleted a significant portion of their energy reserves. So you can also go to section 3, this section in your textbook. It will help you out. So which variable being tested became smaller during cold stress? The answer is, of course, number 4. And in conclusion, what would you say about how fat cells respond to cold shock? And the answer would be increased metabolism and utilize fat reserves. I know that they're increasing metabolism because in the cold stress, the cold shock, if you will, we're seeing an increase in the number and size of mitochondria. Mitochondria's job is metabolism. And then we're seeing a shrinking of the fat cell size, so those fat reserves are being used, so they're not as big a cells as they used to be when they were full of fat. So the take-home message is form follows functions in an organism's cell. Form following function is an important point. Next is lysosomes. You can think of their function as the garbage disposals of the cell. Just like we generate a lot of garbage, a cell does too, and what does a cell do with all the garbage it generates? There are thousands of ongoing reactions of metabolism. They produce a lot of waste that cells must digest and recycle. Many eukaryotic cells deal with this garbage by maintaining hundreds of versatile floating, quote, garbage disposals called lysosomes. These are round organelles. They are filled with about 50 different digestive enzymes and a superacidic fluid. Much like our stomach has enzymes to break down food, and it's acidic to break down food. So the lysosomes are full of a corrosive broth so powerful that if the lysosome were to burst, it would almost immediately kill the cell by digesting all of its component parts. So these are enclosed in their own membrane, which is good because if they're exposed to the whole cell and all that bad stuff leaked out, the cell would die. Tay-Sachs disease is a horrible disease. Look it up. It's genetic. Children with Tay-Sachs are in pain. They typically die by the age of 2. And that disease is a result of messed up lysosomes. Remember how I told you there's 50 different enzymes necessary for the lysosomes to carry out their function? Well, in Tay-Sachs, you have a gene that messes up one of those digestive enzymes because enzymes are proteins, so it messes it up so it doesn't form the right shape, so it can't do its job. And it can't do its job of digesting lipids specifically. It's a lipid digesting enzyme. So all those lipids build up in the lysosome and eventually burst open the lysosomes. And lysosomes then go and kill the entire cell, because remember, it's full of that corrosive stuff, so the cell gets completely withered away when it encounters the lysosome material. So within the first few years of life, this process occurs in large numbers of cells and eventually leads to death, like I said, by age 2. You can read this at your own time. One thing I want to talk about is lysosomes. Just like how cells like liver and muscle that need a lot of energy have a lot of mitochondria, cells that need to digest a lot of stuff would have a lot of lysosomes. So our immune cells, their job is to run around our body, find foreign things and gobble them up and break them down, like bacteria or allergens. So those types of cells would have a lot of lysosomes. The information for how to construct the molecules essential to a cell's functioning and survival is stored on the DNA found in the cell's nucleus. The energy used to construct those molecules and to run cellular function comes primarily from the mitochondria, but the actual production and modification of biological molecules occurs in a system of organelles called the endomembrane system, shown here. The endomembrane system takes up as much as 1/5 of the cell's volume, and it's responsible for many of the fundamental functions of the cell. So remember that lipids are produced within the endomembrane system. Products for export to other parts of the body are modified and packaged here, and amino acid chains are assembled into functional proteins here. Also, many of the toxic chemicals that find their way into our bodies, from recreational drugs to antibiotics, are broken down and neutralized in the endomembrane system. Start with the rough endoplasmic reticulum. It's rough because it has a bunch of ribosomes attached to it, and ribosomes make proteins. So what ribosomes do is they make all the amino acids linked up together to form a protein. And then what the rough ER itself does is it folds those proteins in the correct shape. Remember how proteins have to be in a correct shape to do their correct job. So the ribosomes make the amino acids linked up together to form the linear protein, and then the rough ER makes the protein go into its three-dimensional correct structure. Alright, so remember that the rough ER's primary job is to fold and package proteins that will be shipped elsewhere in the organism. Next is a smooth ER, called smooth because they don't have ribosomes on them. So their job is not to do anything with proteins, really. They are where lipids are synthesized, also where alcohol, antibiotics, and other drugs are detoxified. Alright, so remember lipids would include fatty acids, phospholipids, and steroids. So those are made there at the smooth ER. Another critical responsibility of the smooth ER, especially in human liver cells, is to protect us from the many dangerous molecules that get into our body, such as alcohol, antibiotics, barbiturates, amphetamines, and other stimulants that we may consume, along with many toxic metabolic waste products produced in our bodies. So the smooth ER will detoxify that stuff. You can read this on your own time. Next is the Golgi, and it processes products to transport throughout the body. I like to think of it as the UPS of the cell. You can see all of these little round circles on the microscope image on the right. There's these round circles of different sizes. Those are vesicles. So those are like the packages that are coming into the UPS or getting shipped out of UPS. So those are transport vesicles that bud from the ER and then go to the Golgi, and then in the Golgi they fuse with the Golgi, and then they get shipped back out to where exactly they need to go. So here I'm showing how the various parts of the endomembrane system work to produce, modify, and package molecules within a cell. Alright, which organelle will digest a bacteria that has been phagocytosed by a white blood cell? Phagocytosis just means eating, like a white blood cell ate a bacteria. Which organelle in that white blood cell will digest it? The answer is, of course, number 1, lysosome, because those are the garbage disposals. The cell wall. The cell wall is important for plants. Animal cells do not have them. So now we're going to look at a couple of structures not found in all eukaryotic cells. Because plants are stuck in the ground, unable to move, they have several special needs beyond those of animals. In particular, they can't outrun or outmaneuver their competitors to get more food, nor can they outrun organisms that want to eat them. If they want more sunshine, they have few options beyond simply growing larger or taller. And if they want to resist plant-eating animals, they must simply grow tougher outer layers. The organelle that helps plants achieve both of these goals is the cell wall, a structure that surrounds the plasma membrane. The cell wall is made of cellulose, which is a type of carbohydrate that we do not have in our bodies. The cell wall is nearly 100 times thicker than the plasma membrane, and it gives the cell tremendous structural strength. And that is why it's possible for some plants to grow several hundred feet tall. Cell walls also help to increase plants' water resistance, which is an important feature in organisms that cannot reduce water loss to evaporation by moving out of the hot sun, like we can, and it provides some protection from insects and other animals that might eat them. More take-home messages for this section. Vacuoles, that very, very large, looks like an empty container in the cell. If you look at a mature plant cell through a microscope, like at the bottom right, one organelle, the central vacuole, usually stands out more than all the others because it is so huge and it appears empty. Although it appears to just be an empty sac, it's anything but that. It is surrounded by a membrane and filled with fluid, and it can occupy 50 to 90% of a plant cell's interior. It plays an important role in 5 different areas of plant life. And against that vacuole, you see a lot of those green organelles. Those are chloroplasts. Chloroplasts' job are to harvest the energy from the sun and to make a bunch of sugars. And so what they'll do is they'll deposit those sugars for storage in that vacuole. So nutrient storage is one role of the central vacuole. It stores hundreds of dissolved substances, including amino acids, sugars. Waste management, the vacuole retains waste products and degrades them with digestive enzymes, much like the lysosome does in animal cells. Predator deterrence, the poisonous, nasty tasting materials that accumulate inside the vacuoles of some plants make a powerful deterrent to animals that might try to eat parts of the plant. Sexual reproduction, the vacuole may contain pigments that give some flowers their red, blue, purple, or other colors, enabling them to attract birds and insects that help the plant reproduce. And lastly, physical support. High concentrations of dissolved substances within the vacuole can cause water to rush into the cells through the process of osmosis. The water increases the fluid pressure inside the vacuole and can cause the cell to enlarge a bit and push out the cell wall. This process is responsible for the pressure called turgor pressure that allows flowers and other plant parts to stand upright. The ability of non-woody plants to stand upright is due primarily to turgor pressure, versus like we water a plant readily, all the time, and you see it stand up. That would have good turgor pressure. That means that a lot of water got into the central vacuole and pushed against the cell wall and opened it up. In contrast, if a plant is wilting, there would be a loss of trigger pressure because it does not have enough water in it. You read the take-home messages here. I already mentioned chloroplasts, but they're very, very important. If I had to choose one of the most important organelles in a cell, chloroplast would be a top contender. It's found in all plants, and it's the site of photosynthesis, which we'll talk about in Chapter 4. Photosynthesis is the conversion of light energy into the chemical energy of food molecules, with oxygen as a byproduct. Because all photosynthesis in plants takes place in chloroplast, they are directly or indirectly responsible for everything we eat and for all the oxygen we breathe. Life on Earth would be vastly different without the chloroplast. I don't think you need to know these parts of the chloroplast. I just want you to know that this is where photosynthesis occurs, and we'll talk more about them in the chapter. The chloroplast is another organelle, much like the mitochondria, that supports endosymbiosis theory. And that's because they resemble photosynthetic bacteria. So not only plants undergo photosynthesis, but some bacteria can too. They have circular DNA and that double bag within a bag membrane structure. So according to endosymbiosis theory, the chloroplasts remained alive, and rather than becoming a meal, they became the cell's meal ticket, providing food for the cell in exchange for protection. So here's a review of all the parts of a cell and their functions. I suggest that one way to study is to make this chart, fill in the structures on the left, and then see if you can, from memory, do those check marks and write out the functions. I would try and do that from memory, and then once you've filled in as much as you can, go back to your notes in your book and your slides and completely fill in that chart. And then I would suggest the next day you do it again until you can 100% fill out this chart from memory. So which function is specific to the chloroplast? The correct answer is number 4. So 1, used for detoxification, that would be which organelle? The smoothie R. Number 2, the garbage disposal of the cell would be which organelle? The lysosome. Used to maintain turgor pressure, that means all the water storage, that would be which organelle? The central vacuole. And then chloroplasts convert light into chemical energy. And 5 converts chemical energy into ATP, was that mitochondria, where respiration occurs. Alright, so we're ready for Chapter 4. This whole chapter is about energy. So, from the sun to you in just 2 steps. Here's our learning objectives. Those are the sections that I'll be lecturing on that you are responsible for knowing in your book. Specifically, the objectives are to understand and explain how energy flows from the sun through all life on Earth. You're going to have a writing assignment on that specific learning objective. I'll be posting that soon this week with an explanation in it, so don't worry about it. Next is how photosynthesis uses energy from the sun to make food, and then how cellular respiration converts that food into ATP, which is a universal source of energy. And then we'll talk briefly about alternative pathways to energy acquisition besides respiration. It's imperative to know and understand and appreciate that energy flows from the sun and then through all life on Earth. All of the energy, all of it, that's all originating from the sun. So imagine that you are on a long road trip. Your car's fuel gauge is nearing empty, so you pull off the highway. Instead of driving to a gas station, however, you head to the back of a fast food restaurant and fill the fuel tank with used cooking grease. You head back to the highway, ready to drive several hundred miles before needing another pit stop. Many vehicles run on biofuels, which are fuels produced from plant and animal products. Most vehicles, however, run on fossil fuels still. An example is gasoline. These fuels, which also include oil, natural gas, and coal, are produced from the decayed remains of plants and animals modified over millions of years by heat, pressure, and bacteria. So regardless of if we're talking about biofuels, fossil fuels, or food fuel like what we eat, they are all chemically similar. They're chains of carbon and hydrogen atoms, and those chains are contained of bonds, chemical bonds between those atoms, and the energy we get is stored in the bonds. Is it surprising that they are all similar? It shouldn't be, because energy from the sun is the source of all energy stored between chemical bonds in the atoms in all these fuels. In this chapter, we explore how plant, animal, and other living machines run on energy stored in chemical bonds. These are things we touched on in Chapter 2, so they're coming back. Energy capture and conversion occurs in two important processes that mirror each other. One is photosynthesis. That is a process by which plants capture energy from the sun and store it in chemical bonds of sugars and other food molecules they make, mostly sugars. And two, cellular respiration, the process by which all living organisms release the energy stored in the chemical bonds of food and use it to fuel their lives. So here, this chart is showing you that the sun is the original source of all energy. Photosynthesis in plants or photosynthetic bacteria then capture energy from the sun and store it in the chemical bonds of sugars. Then those plants and other animals are going to do cellular respiration. Don't forget that plants also do cellular respiration. So they do photosynthesis and respiration. So what respiration does is it releases the energy stored in the chemical bonds of food molecules they eat in the case of animals, or that they produced themselves in the case of photosynthesis, and use it as fuel for energy. So here's the take-home message. Remember, the breaking of bonds is where all the energy comes from. And there are two forms of energy. Humans, plants, and other living organisms need energy for their activities, from thinking to moving to reproducing. So energy is the capacity to do work, and then work itself is defined as moving matter against an opposing force, right? Could be you pushing something up a hill. That would be work of you moving, let's say a rock, a matter of rock, against an opposing force, which is gravity pushing down on the hill. There are two types, kinetic on the left and potential energy shown here on the right. Kinetic energy is the energy of motion, or another way of saying that is moving objects. So legs pushing bike pedals and flapping of wings of a bird are examples of kinetic energy. Also, heat energy is kinetic energy. It results from lots of molecules moving rapidly. And because it comes from the movement of high energy particles, light is also a form of kinetic energy. Probably the most important form of kinetic energy on Earth, because that's the sun, and we use that sunlight, as I said before. Then there's potential energy, which is energy stored in an object. Concentration gradients are potential energy, and then food has potential energy stored where? Stored in those chemical bonds, because then they break and we have kinetic energy, but when they're still in a bond, that's potential energy. An apple has potential energy, as does any other type of food. Why? Because the chemical energy, again, is stored in the chemical bonds. Making up the food can be broken, and the energy released during cellular respiration, enabling you to run, play, and work. So the energy stored in bonds is what type of energy? The answer is, of course, potential. The next section is talking about thermodynamics. So as energy is captured and converted, the amount of energy available to do work decreases. So this is important to know, kind of fascinating, that only 1% of the energy released by the sun that the Earth receives is captured and converted by plants. So only 1% of all of that sunlight energy is converted into chemical bond energy in food. So what is going on with the other 99%? Some of it is reflected back into space or absorbed by the land, oceans, and atmosphere, but most of it is transformed into heat. So that's shown down at the bottom. Like on the bottom left, you have the light energy from the sun. Some of it, a little bit of it, is going into chemical energy in plants, but a lot of it's going off up in the air in those squiggly lines in the form of heat. Also, when you have on the right, when you have chemical energy stored in those bonds and then you break them, energy is going to be lost as heat as well, kind of like when you work out. A lot of heat is given off, and only a little bit is used directly for kinetic energy of forward motion. This is because of the first law of thermodynamics. The first law of thermodynamics states that energy can change form but never disappear. And that's an important feature of energy in the universe, whether we are looking at the sun and the earth or a human and a rice bowl. Just as energy can never disappear or be destroyed, energy can never be created. All the energy now present in the universe has been there since the universe began. And everything that has happened since then has occurred by the transformation of one form of energy into another. So in all of our eating and growing, driving and sleeping, we are simply transforming energy. So if you could think of any other examples, that would be good for you to test yourself. Whether it's a person converting the chemical energy in a plate of spaghetti into the kinetic energy of running a marathon, or a car transforming the chemical energy of gasoline into the kinetic energy of forward motion, some energy is converted to heat, which is the least usable form of kinetic energy. For example, in automobiles, about 3/4 of the energy in gasoline is completely lost as heat. So you could think of it, for every $10 spent on gas, only about $2.50 actually goes towards moving your car. Although heat is certainly a form of energy, remember it's kinetic energy, it is not easily harnessed to do work. It is almost completely useless to living organisms for fueling their cellular activity. Put another way, the second law of thermodynamics tells us that although the quantity of energy in the universe is not changing, its quality is. Little by little, the amount of energy that is available to do work decreases. So the amount of energy available to do work decreases. Now that we understand that organisms on Earth cannot capture every single bit of energy released by the sun, and that energy conversion is inefficient, we can look at the chief energy currency of the cell, which is ATP. And you can think of ATP molecules like free-floating rechargeable batteries for your cell. So how do cells directly fuel their chemical reaction? Light energy from the sun is not directly used to fuel cellular work. It's used to make food, and then that food is used in respiration to make ATP for cellular work. Alright, so first it must be captured in the bonds of a molecule called ATP, or adenosine triphosphate. That tri stands for 3, and that's because ATP has 3 phosphate groups. I boxed it here in red. ATP is a simple molecule. It's composed of an adenine, a ribose sugar, and then phosphate groups. I just want you to really focus on knowing the phosphate groups. Because the ATP is what makes it, the phosphate groups are what make ATP so effective in carrying and storing energy for a short time. These phosphates are negatively charged, so they're not stable. Like, they don't like being next to each other. Because of that, there's a lot of energy stored in the bonds between those phosphate groups. The instability of these 3 high-energy bonds makes the 3 phosphate groups like a tightly coiled spring or a twig that is bent almost to the point of breaking. So there's a lot of potential energy there. So with the slightest push, one of the phosphate groups will pop off, releasing a little burst of energy from that bond that it broke, that the cell can then use to do work. Okay, so that's what I boxed here in red. ATP on the left has those 3 phosphate groups with those high energy bonds between those phosphates. And then a phosphate group will come off, releasing energy. You then have an ADP, or an adenosine diphosphate, because now there's only 2 phosphate groups. So how is it rechargeable? We can then add that phosphate group back on to ADP to make ATP, and then we can use that ATP again to do work. It's summarized here. Alright, so here's the recycling part. So each time a cell expends one of its ATP molecules to pay for an energetically expensive reaction, a phosphate is broken off and energy is released. What is left is a molecule with 2 phosphates called ADP and a separate phosphate group labeled in this diagram as just PI. That's a phosphate group. An organism can then use ADP and the free-floating phosphate as well as an input of kinetic energy to rebuild those ATP stocks. The kinetic energy is converted to potential energy when the free phosphate group attaches to the ADP molecule and makes ATP. In this manner, ATP is like a rechargeable battery. So here is the ATP story in a nutshell. Breaking down a molecule of sugar in a glass of orange juice, for example, leads to a miniature burst of energy in your body. The energy from the mini explosion is put to work building the unstable high energy bonds that attach phosphate groups to ADP molecules, creating new molecules of ATP. Later, perhaps only a fraction of a second later, when an energy consuming reaction is needed, your cells can release the energy stored in the new ATP molecules. Alright, so now we understand ATP and the whole cycle overall from the sun all the way to the ATP. We're going to talk about the 2 processes between the sun and the forming of the ATP. Those are photosynthesis and respiration. Photosynthesis, again, uses energy from sunlight to make food. I just want to point out here that although plants are the most visible organisms that have evolved the ability to capture light energy and convert it to organic matter through photosynthesis, they are not the only organisms capable of photosynthesis. There are bacteria and unicellular eukaryotic organisms also capable of using energy and sunlight to produce organic materials. Alright, I want you to know the big picture. I want you to know the inputs and the products. There are 3 inputs to the process of photosynthesis. 1, obviously light. 2, carbon dioxide. This comes from the atmosphere. And 3 would be water. This would come from the ground. From these 3 inputs, the plant produces sugar and oxygen. So the 2 outputs are sugar and oxygen. If we did not have plants, we'd have a lot of carbon dioxide in the air and very little oxygen. Alright, so think of it like this. Photosynthesis. So photo means like a photograph capturing light. So photo, capturing light, and then synthesis, making something, synthesizing sugar. Sometimes it helps me if I can break down a word to its root parts. Photosynthesis takes place in those organelles called chloroplasts that we talk about. This is why leaves are green. It's because the cells near the surface are packed full of chloroplasts. At the surface, it can get the most light, so that's where there's the most chloroplasts there. It makes it possible for the plant to use the energy from sun to make sugars, which is their food, and then we eat them and use that as our food too. And that's all I want you to know about that section. I'm not probably going to test on this so much, but I do want you to understand overall that light energy travels in waves. And then there's pigments inside of plants that absorb specific wavelengths of light. So if you've ever seen a rainbow, it goes from red, Roy G. Biv, so red, orange, green, blue, yellow, violet. You see all those colors in the rainbow. Each one of those colors is a specific wavelength of light. Alright, so it's a type of kinetic energy, and there's these little energy packets called photons in those wavelengths of light. So when plants use sunlight energy to make sugar during photosynthesis, they use the visible portion of the electromagnetic spectrum, which is light. So just know that they use light. In pigments. So pigments are in the chlorophyll, chlorophyll are in the chloroplast, which is the whole organelle. Alright? So the organelle is a chloroplast. It's full of chlorophyll, which is what makes it green, and which is what actually absorbs the light molecules, the photons of energy in the light. So chlorophyll is the pigment molecule in plants that absorbs light energy from the sun. Chlorophyll molecules are embedded in the chloroplasts, which are found primarily in plants' leaves. So just as light energy excites electrons in the pigments responsible for color vision in humans, electrons in a plant's chlorophyll can become excited by specific wavelengths of light and capture a bit of this light energy. Alright, so there's different pigments. There's several listed here. Mostly they're made of chlorophyll. There are these some of these things called carotenoids, but we'll just focus on chlorophyll being the pigment inside the chloroplast. And remember I said light is different wavelengths? See at the bottom of that rainbow of light? Chlorophyll absorbs light in that blue range and in that orange red range. So it does not absorb light in the green range. This is why plants are green. The green wavelengths of light are not absorbed by the chlorophyll pigments inside of the chloroplast of plant leaves, and therefore they are reflected back. So what we're seeing is the reflection of that green light that is not being used for photosynthesis. Okay, so here is a summary of that. Alright, remember, photosynthesis uses the captured energy of sunlight to make its food. But then cellular respiration will convert food molecules into ATP. Alright, so notice you need to know the differences between respiration and photosynthesis and how they work together. Alright, so now respiration. Remember, this takes place in plants and animals. Food is fuel, and all the activities of life, growing, moving, reproducing, require fuel. Plants, most algae, and some bacteria obtain their fuel directly from the energy of sunlight, which they harness through photosynthesis. But less self-sufficient organisms like humans, alligators, insects, any other animal, must get its energy from the food it eats. Okay, so plants make their own food, but we got to go eat food. This energy comes from the photosynthetic organisms, either directly from eating those plants or indirectly from eating animals that eat plants. All living organisms extract energy from the chemical bonds of molecules, which we are saying are food, through a process called cellular respiration. This process is a bit like photosynthesis, but in reverse, sort of. Not all the details are the same in reverse. But anyway, in photosynthesis, the energy of the sun is captured and used to build molecules of sugars such as glucose. In cellular respiration, though, plants and animals break down the chemical bonds of sugars and other food molecules such as fat and proteins to release the energy that went into creating them. So that's the backwards part. Alright, human example. We eat food. What is it? It could be a plant. Where did it come from? Could be a buffalo. But all of that food came from the plant, which made it from the sun. We then digest that food. We'll take the case of lettuce. We break it down. We then get all these nutrients into the bloodstream, like glucose from digesting the plant. We get all the glucose out that it made through photosynthesis. And then all that glucose nutrient molecule will go to the cells for respiration. So when a food molecule has been completely processed, the cell has used the food molecule's stored energy along with oxygen to create a large number of high energy storing ATP molecules, water, and carbon dioxide. All right, the inputs for photosynthesis are the answer is B. So photosynthesis input would be, write this equation down. Photosynthesis equals sunlight plus water plus carbon dioxide, arrow pointing to the right, oxygen plus sugar. And then respiration is sugar plus oxygen, arrow to the right, carbon dioxide plus water plus ATP. So that's all the inputs and outputs for photosynthesis and respiration. You should memorize those. There they are. I'm not going to go through all the steps of respiration. Just know the first step is glycolysis, which means lysis is the splitting, lysis of sugar, glyco. I bring that up because in chapter 2 we talked a lot about carbohydrates and glucose in our diets in general. So the first step of respiration is breaking down glucose molecules. Right? You don't need to know all these steps on the left, but on the right I boxed all of the outputs that you get. So this is where you get the ATP, the carbon dioxide, and the water in that equation. There all are alternative pathways to energy acquisition. So far we just talked about respiration, but not every organism does respiration. And sometimes we don't even do respiration. So during strenuous exercise, our muscles can require more oxygen than is available to them. So if you don't have oxygen, you can't do respiration. Oxygen is one of the required inputs for respiration. So if you run or swim as fast as you can, you'll feel a burning sensation in the muscles. So what is happening is that your muscle cells are suddenly becoming acidic, and the acid buildup occurs when we demand of our bodies bursts of energy beyond what they can sustain through respiration. So with rapid strenuous exertion, our bodies soon fail in delivering oxygen from the lungs to the bloodstream, which then goes to the cells and then goes to the mitochondria for respiration. That oxygen deficiency then limits the rate at which the mitochondria can break down fuel and produce that ATP to do work with. Organisms, though, aren't stopped for too long because of oxygen lack. What they do is they have a backup method for breaking down sugars. So in the absence of oxygen, we can break down pyruvate. You don't need to know that, but I need you to know there's alternative pathway to respiration and why. And yeast also does this. So they don't need oxygen. Yeast is important for fermentation to make things like alcohol and bread. So fermentation is a process by which cells obtain energy in the absence of oxygen. So we're most familiar with it with yeast, but our bodies do it too, and we get that lactic acid buildup if you ever work out for a long time and you don't get enough oxygen. We've talked a lot about carbohydrates from plants, and I don't need you to know all these details, but I do want you to know, revisiting chapter 2, that we eat fats and proteins in addition to carbohydrates, which we talked about, right? So we don't just eat plants, we also eat animals, which give us, in addition to carbohydrates, those fats and proteins, especially fats from animal products. And since I'm not going through the details of respiration, I can't really go into the details of how we use fats and proteins, whereas I showed you carbohydrates would start with glycolysis, breakdown of those glucose, which make up the carbohydrate. But just know that you can indeed use sources of food besides carbohydrates, and we talked about those in chapter 2. Alright, these are the things I expect you to know is, again, how energy flows from the sun through all life on Earth. Know about those two types of energy, kinetic and potential, and you need to know that the energy is stored in chemical bonds, and the chemical bonds are potential energy. I want you to know how photosynthesis uses energy from sunlight to make food. I need you to know where it happens in the cell, so that would be the chloroplast, and the inputs and outputs, right? So what goes into the chloroplast to make photosynthesis and what comes out at the end of photosynthesis. I want you to know how cellular respiration converts food molecules into ATP, and that ATP is a universal source of energy. I want you to know where it happens, which is the mitochondria. I want you to know the inputs and outputs of respiration. Also know that there are alternative pathways to energy acquisition. The one we covered was fermentation, and that happens because there's not enough oxygen around. Okay, thank you. Make sure that you are doing your discussion forum questions, turning in your homework and your quizzes on time.